The overall goal of this procedure is to rapidly and accurately measure flight performance in Drosophila. This is accomplished by first dropping flies into a flight cylinder. The second step of the procedure is to image the landing surface with a digital camera. The final step is to analyze the data using imaging software by measuring the landing height of each fly. Ultimately, this procedure helps identify mutations and conditions such as aging or neurodegeneration which impair flight ability and helps to quantify flight performance.
The main advantage of this technique over existing methods, such as the original flight tester, is that it's more appropriate for high-throughput screens for mutants and other conditions that impair flight.
We first had the idea for this method when we realized how much time was involved with testing a large number of genotypes using the original flight tester.
Secure the flight cylinder to a ring stand using chain clamps. Leave approximately 3 centimeters underneath the cylinder for the weigh dish. Load the weigh dish with a thin layer of mineral oil and slide it under the flight cylinder. Secure the funnel to a second ring stand. Adjust the height of the funnel so that the bottom of the funnel is flush with the top of the flight cylinder. The narrowest part of the funnel must be narrower than a fly vial. The diameter of the drop tube should be slightly wider than a fly vial, so the vial is able to drop freely. Insert the drop tube into the top of the funnel and secure it using a claw clamp. Next, from a polyacrylamide sheet, cut a rectangular piece the length of the flight cylinder with a width slightly